All right. Hi there. Welcome to the homework review for chapters 1 and 2 of Statistics CJ 613. And I'm John DiCarlo, and I'll be walking you through the questions and answers for the review. So let's take the first one, and it's a pretty important one, actually. The question is to find the terms parameter and statistic, and be sure that the concepts of population and sample are included in your definitions. So I just wanted to read the question to you overall first. A parameter is a characteristic of usually a numerical characteristic that describes a population. Okay, and so a parameter goes with population. And I'm just going to grab my felt tip pen here. So parameter goes with population. Notice that they are both in green and they both start in P. That's the mnemonic for you. Parameters go with populations. A statistic is a characteristic that usually describes a sample. So just remember that statistic begins with S, sample begins with S. They go together like a horse and carriage, and that's from the song, by the way. Statistic and sample are linked. Let's talk real quickly to review about what a population and a sample are. Let's say that we wanted to do an experiment and we needed some people to experiment with on their fear of crime. Okay, so our population here in Connecticut is about 3.5 million people. Obviously, it would be really expensive to do a survey and distribute it to 3.5 million people. 3.5 million surveys would be a huge amount. So we would take a representative sample. Remember that word sample? And we would say maybe about 500 people, and we would send our sample to about 500 people, randomly selected people. And it's important that a sample be randomly selected. We'll talk about other ones, and for those of you who have had research methods with me, you remember about stratified samples and snowball samples, etc. Those are perfectly legitimate, but for a true experiment, we want a random sample, okay? People who are just selected at random. And so a sample is just a subset of a population. Okay, good. Let's pop over to the next question, which is question 4. A researcher plans to compare two treatment conditions by measuring one sample in treatment 1 and one sample in treatment 2. Okay, the researcher will then compare the scores for the two treatments if there is a difference between the two groups of scores. Okay, and it's asking us to briefly explain how the difference may have been caused by the treatments, number 1, and number 2, or B, briefly explain how the differences simply may be sampling error. So let's take a look. If treatments 1 and 2 affect individuals differently, then the scores in the first sample will be different from the scores in the second sample. So let's say we're testing heart medicine, okay, and heart meds, and we have heart med A and heart med B, and we have a sample of 20 people in each group, and we give heart med A to the first sample and heart med B to the second sample. And we have less heart conditions, fatal heart attacks, in A. That means that that's working differently. So we're correlating the heart med A with less heart attacks. Okay, so that is how they are different. The scores are actually different. And B, it's saying briefly explain how the differences simply may be sampling error. Well, let's say that we gave everybody the two groups A. We're still going to get different means. Okay, even if they have the same treatment, we're still going to get different means because let's say that 3.5 million people in Connecticut again who have heart conditions would give us maybe more exactly the same numbers for the two groups. But because we are not dealing with a population, we're dealing with, don't forget, a sample, then we're going to get something called sampling error. Sampling error is simply the difference between our sample and a whole population. As the sample gets bigger, we have less and less sampling error, or sample error, if you will, until we get to the size of the whole population, at which point there is no sampling error because we don't have a sample anymore. We have a population. So let's move on to the next question. And I hope that you understand that. We're going to pop over to the next one here, and that is question 8. It says that Downs and Abwender found neurological deficits in soccer players who are routinely hit on the melon with soccer balls, as compared to swimmers who are also athletes but who are not regularly hit in the melon. Is this an example of an experimental or a non-experimental study? What do you think? Well, it's not an experimental study because there is no independent variable, all right? They're just comparing two pre-existing groups of individuals. They're correlating, if you will. It's a correlational study rather than an experimental study. They're just comparing two groups. They're correlating banging your head with a ball with neurological deficits. So what do we need to have an experiment? We need some variables. What kind of variables do we need? We need two kinds. Okay, we need an independent variable, an IV, and we need a dependent variable, or a DV. By manipulating my independent variable, I make, or hopefully make, the dependent variable change, and that is called manipulation, experimental manipulation. And let's say that I manipulated the amount of, here's a good one. Let's say I have two groups of plants, and I want to experiment on the effect of water, H2O, on plant growth. And so I very carefully measure out and give different amounts of water to my two groups of plants. So my water is my independent variable. My dependent variable, what is it, guys? It's the plant growth. Okay, it depends on how much water they get, or how the independent variable, the water, manipulates the growth, or the dependent variable. And what I want to do in an experimental situation like this is I want to make sure I control for other variables that might also get in my way, or what we call in the biz confound. Okay, where's my pen? Confound my results. It's F-O-U-N-D, and confound my results. So let's say that inadvertently I gave the two experimental sample groups different amounts of sunlight. Do you think that would affect the growth rate also? Sure it would. So I would have accidentally introduced a confounding variable, the sunlight, into the experiment. So I must control for those types of variables. And in the plant experiment, I would want to give each group of plants the exact same amount of sunlight. Okay, popping over to the next one. Just going down here. I've lost my mouse, sorry. Okay, now let's take a